Okay, now, lecture number 17, we're going to start moving into chapter 28 content. We will not be covering nearly as much detail from chapter 28 as we did with chapter 27, so keep that in mind if you're reading along with your textbook. But we have been through what it takes to produce sperm, what it takes to produce functional oocytes, and we've talked about the ovarian and the uterine cycles and the hormonal controls in males as well. And so finally we're ready to fertilize an egg. So that's going to be the subject of this lecture. All right, some timing things here first. All right, so pregnancy, the definition of pregnancy. This includes all of the events from fertilization, when sperm meets the secondary oocyte, until the infant is born. The gestation period, actually the time is different from the actual pregnancy period. A lot of people don't know this. But the gestation period, which is about 280 days on average, obviously everybody knows that varies from pregnancy to pregnancy. It's actually timed from the beginning of the last menstrual period until birth. So really, so if you think about what we learned about in the previous lecture, ovulation does not occur until about 14 days into the ovarian cycle. The menstrual period starts at day 1. Okay, so the timing of a pregnancy, when a woman says I'm so many weeks pregnant, that's actually including the 2 weeks prior to ovulation taking place. Then ovulation takes place around day 14, and as we'll see, the secondary oocyte that gets released out of an ovary has to be fertilized by a sperm very quickly. So it's going to happen right around, within about 24 hours or so after ovulation takes place. So keep that in mind. If a woman says, I'm 1 month pregnant, I'm 4 weeks pregnant, in reality, fertilization happened 2 weeks ago. Or if you're 8 weeks pregnant, fertilization actually occurred just 6 weeks ago. So you have to kind of keep some of those timing things in mind. Okay, some additional terminology. Here you're seeing some of the stages of development and the changes in size and so forth that occur pretty quickly. So from the time of fertilization, 3 weeks later, a developing embryo looks like this. Here's 5 weeks, here 8 weeks, and then there's a 12-week fetus, which has already developed a pretty good size. Another term, the conceptus, is another term for the developing offspring. An embryo is the conceptus from fertilization through week 8. And then the fetus is what the conceptus is called from week 9 through birth. All right, so again, what is fertilization? This is when the sperm's chromosomes, so 23 of those, combine with the chromosomes of an ovum, 23 of those. You put 23 together and 23 together, and now you have 46. And so that is a diploid cell, and that is the first cell of the new individual, and that's also called a zygote. The zygote is the first cell of the new individual. And as I've mentioned before, there's a very small window of opportunity for this to take place. So ovulation occurs, the secondary oocyte enters the fallopian tube, and it's actually only viable for 12 to 24 hours after ovulation takes place. Now the sperm in general are viable for a longer period of time after ejaculation. So they may have been ejaculated into the female a day or two in advance of ovulation, and they still may be able to fertilize that secondary oocyte, because they'll generally live about 24 to 48 hours after ejaculation occurs. All right, so if you're trying to get pregnant, coitus, sexual intercourse, must occur no more than about 2 days before ovulation or 24 hours after ovulation. So you've got that small window of opportunity, and fertilization is generally going to occur in a fallopian tube. So here's a diagram from your textbook. So the semen, of course, gets deposited right in here at the opening to the cervix, the external os of the cervix, and then they're going to swim up into the uterus, and also the uterus contracts. You have peristalsis that takes place as the smooth muscle in the wall of the uterus contracts, and that helps propel these sperm cells forward. And then they start swimming their way up at the fallopian tubes, and meanwhile, here's a secondary oocyte that got ovulated out. It passes through the infundibulum into a fallopian tube, and somewhere in here you're going to have fertilization occur. All right, so taking a look at that, why does it take so many sperm? Why do men have to produce millions and millions and millions of sperm cells for each ejaculation? Well, just looking at what sperm cells have to accomplish to find a secondary oocyte, it's probably not a big surprise, but millions will leak out of the vagina immediately after ejaculation. So even though semen, we talked about how it coagulates right after ejaculation, but then it has to reliquefy so that the sperm can actually swim out and start their journey. Nevertheless, a lot of them are going to leak out. The vaginal environment is not particularly friendly. The vaginal lining has a lower pH than you would typically find in body fluids. And we did talk about how semen has alkaline compounds in it that help counteract that acidic pH. So that just does enhance sperm survival. But nevertheless, a number of sperm are probably still going to wind up dying along the vaginal lining because of the unfriendly conditions that are there. And then, as you might imagine, not all of them are going to make it into the cervix. And then even when they get into the uterus, phagocytes like macrophages may be positioned in the uterus, and they see these sperm cells as things that are foreign to the female's body, so they're going to phagocytose them. So when you consider all these things, it's a miracle that any of these sperm cells ever make it to a uterine tube or fallopian tube in the first place. And the numbers are thought to be down into the thousands that actually make it to both uterine tubes. All right, so you've got those lucky thousands that actually make their way up through the fallopian tube. And then what is actually involved with fertilization? One thing that's very important is you just want one sperm to fertilize the egg. I'm having trouble writing here with my stylus on my iPad. You want one sperm to achieve fertilization. All right, so this big giant cell we're taking a look at here on this diagram from your textbook, this is a secondary oocyte. So that secondary oocyte has not finished meiosis II. And here you see sperm cells coming in. They've found the secondary oocyte. They're going to try to fertilize it. And what they're doing here, right, here are the cells of the corona radiata, those granulosa cells that stayed with the secondary oocyte at ovulation. And the sperm cells can kind of wiggle their way through the granulosa cells. Then they reach this darker yellow border there. That's your zona pellucida. And if you remember, that is a glycoprotein covering that protects the secondary oocyte. And the sperm cells cannot easily penetrate that. They have to work to make their way through the zona pellucida. So when the sperm bind to the zona pellucida, remember on the head of the sperm cell, there's the head. So you got the DNA, the chromosomes are in there, and then it kind of has that helmet-like structure. Okay, and that's called the acrosome. The acrosome, that little covering over the head of the sperm cell, contains a lot of enzymes, acrosomal enzymes, and those acrosomal enzymes, like you're seeing here on the diagram, they degrade the zona pellucida. So they digest, they break apart the glycoproteins that are present in the zona pellucida. Now once they make their way through there, the secondary oocyte, these little Y-shaped molecules that you see there on the diagram, those are sperm cell receptors. So they kind of function like hormone receptors. They're very specific for binding the heads of sperm cells. So finally, one lucky sperm cell works its way completely through the zona pellucida, and it attaches to a sperm cell receptor. So it's going to be our winner. And after that happens, the membrane around the head of the sperm and the membrane of the secondary oocyte, they fuse together, they kind of melt together, and that releases the nucleus of the sperm inside the secondary oocyte. So what prevents other sperm from getting in there? There are two things here. They're summarized at the bottom of this diagram here. As soon as one sperm cell attaches to a sperm receptor and the nucleus of the sperm enters inside there, there's a rise in calcium levels inside the secondary oocyte that's being used like a second messenger, kind of like we learned about that at the beginning of the semester with hormones that bind to hormone receptors on the cell surface, and that sets off an instantaneous set of reactions, and one of those is called the cortical reaction. And what that does is it triggers changes in the zona pellucida, and it hardens the zona pellucida, and it makes it impossible for additional sperm to digest their way through, or much more difficult for them to do so. And then the other thing that happens is there are enzymes that clip off those remaining sperm receptors. So that's another reason why no additional sperm will be able to fertilize this secondary oocyte. So those things are supposed to safeguard against polyspermy. That would be where the nuclei of two sperm cells made it into a secondary oocyte at the same time. If that happens, you're going to have an inappropriate number of chromosomes from the male that wind up inside this oocyte, and that's not going to be a viable embryo. So there will be no embryo that would develop in that particular case. This is a diagram from your textbook which is showing you what happens, okay, after the sperm penetrates the secondary oocyte. Then you finish meiosis 2. And again, meiosis 2, so you still have inside this secondary oocyte 23 duplicated chromosomes from the mother. And to complete meiosis 2, those duplicated chromosomes have to separate. And one set stays with the ovum. We now have an ovum. During meiosis 2, you go from the secondary oocyte to an ovum. And the ovum has one set of 23 chromosomes. The other set get dumped into the second polar body. If you guys remember us talking about that before. All right, and then if you look in here, the nucleus of the sperm cell is in there, and it has the 23 chromosomes from the male. The nucleus from the ovum that we now have are located in there as well. And the nuclei swell. The nuclei approach each other. Here on the diagram from your text, they're talking about the mitotic spindle forming between them, and that's the apparatus that is going to help achieve mitosis. So for this embryo to develop, there's going to be lots and lots of mitosis, where you just go from one diploid cell to 2, 2 to 4, 4 to 8, 8 to 16, etc. So we're back to doing regular old mitosis instead of meiosis. Keep that in mind. Meiosis is a special process that cuts in half the number of chromosomes that you have in gametes, sperm and ova. Okay, so eventually the chromosomes from the male and the female meet up with each other. So now you have two copies of each. You've got two copies of chromosome number 1. One came from dad, one came from mom. Two copies of chromosome number 2. One came from mom, one came from dad. And don't forget also, you're going to have two X chromosomes or an X and a Y. So mom is always going to provide an X chromosome, because females have two X chromosomes. They don't have a Y chromosome to, to provide. The sperm cell will have either brought in an X or a Y. So if the sperm cell brings in dad's X chromosome, then this is going to turn out to be a female embryo. If dad's sperm cell brought in a Y chromosome, then you're going to have an XY embryo, and that's going to be a male. Okay, once the chromosomes have combined from the mom and dad, you have the first cell of the new developing embryo, and that's called a zygote. That is cell number 1 of the new embryo. This is a huge cell. So again, if you look, we've talked about how these oocytes are big, gigantic cells, and all the cytoplasm and all the organelles, other internal contents of that giant cell are still there to help out this first cell of the new individual that is forming. All right, and then what happens next? So here's your first cell of the new embryo or new individual. That's called a zygote. It's going to start doing mitosis. So 1 cell goes to 2, 2 goes to 4, 4 goes to 8, 8 goes to 16, 16 goes to 32. And it takes a while for human cells to divide. They have to undergo mitosis. We didn't go through all the details of mitosis, but it's a fairly complex process. So 2 days after fertilization, our little embryo here only has 4 cells. It's at a 4-cell stage. And notice where it is. It's still traveling through the fallopian tube. Also notice that those 4 cells there, their combined sizes are the same as the original single cell of the zygote. So at first, when cell division is taking place, it's like the one giant cell gets split into 2 cells that are half the size of the original. And then those 2 get split into 2 cells that are now 1/4 the size of the original. So the individual cells get smaller and smaller and smaller at first. About 3 days after fertilization, you have a morula. So that is a solid cell mass. And then as our developing embryo is making its way down here into the uterus, it takes on this type of configuration. This is called a blastocyst. That's the name for the embryo at this stage. And if you notice here, you've got a mass of cells right there, and then you've got another mass of another covering of cells around the outside. And then you have space. You have a blastocyst cavity there in the center. So the way to visualize this is imagine you like have a tennis ball. You guys know the tennis ball is hollow on the inside. So you have cells around the surface of the tennis ball that are going to eventually develop into structures that support the developing embryo, things like the amniotic membrane for the amniotic cavity. And then you have this little cell cluster inside. That's called the inner cell mass. And the inner cell mass, those cells are actually going to develop into the embryo itself. That's going to become your new human individual. Now, all these cells originated from the zygote. So genetically, they all have the same DNA. They have the same set of genetic blueprints. But already, even in this very early embryo that's developing, the cells are beginning to take on different jobs, which means some cells are reading some genetic recipes, others are reading other genetic recipes on the DNA, and they're beginning to take on some of their special tasks. So as we're going to see, these cells that are around the surface of our blastocyst, they're going to become the structures that will support this embryo and eventually a fetus that's developing inside the womb. The inner cell mass there, those are the cells that are actually going to develop into the embryo itself. All right, implantation. So finally we're ready to implant into the endometrial lining of the uterus, and that begins about 6 to 7 days after ovulation. Remember, fertilization is going to have to take place pretty quickly after that. The trophoblast cells adhere to the endometrium and proliferate. So if this is our blastocyst here, And here's our inner cell mass right in there. These cells that are forming the surface of this ball, this hollow ball, those are called your trophoblast cells. That's where they're going to be located. And so they're the ones that are actually going to have to stick to the endometrium and start invading the endometrium to allow this very early embryo to implant. Still called a blastocyst at this point. That's just referring to this hollow ball that you have with the inner cell mass on the inside. Implantation takes a while for our little teeny tiny embryo to accomplish, but it is completed by about the 14th day after ovulation. So we're about 2 weeks now into the development of this embryo, and still very, very tiny, and it has embedded itself into the lining of the endometrium. So this diagram from your textbook is showing this taking place. Okay, so this is the endometrium, the inner lining of the uterus, and it's showing you a couple of the different cell types that you would find here. And here's our blastocyst. This is our very early embryo. And so these trophoblast cells you see here at the surface, and keep in mind this is a 3D structure, they attach to the endometrial lining. And then the cells that made contact, oops. Come on, pen, come back up. The cells that made contact here, so these are cells of the embryo right in there. They're those trophoblast cells. The ones that made contact with that endometrial lining, they start dividing over and over. They're doing mitosis, and they begin invading, digging into, they start creating these little projections that stick out and embed themselves in the endometrial lining. And again, the cells that are here in this cluster, that's your inner cell mass. That's going to be your future embryo. All right, so that concludes that lecture. The next one, lecture number 18, we're going to talk a little bit more about hormones again, because we need to return to the hormone story to talk about hormonal changes that will help us maintain the pregnancy. And then we're also going to talk about placentation, the development of the placenta, which forms from the endometrial lining of the mother, and then structures that develop from these trophoblast cells in that super early embryo. Those things are going to combine together to form the placenta, and the placenta, of course, is the organ or the structure that sustains the developing embryo and the fetus for the rest of the pregnancy. It's going to be the structure from which oxygen gas can be sent from the mother's blood into the blood of the developing embryo and fetus, and it will also send nutrients over, takes away wastes out of the blood of the developing embryo and fetus. So we'll talk about how that structure develops in lecture number 18.